What's going on, everyone? If you like this video and want to support the creation of more content on this YouTube channel, don't forget to hit like and to subscribe. It's just a small thing, but it goes a long way to helping this grow. I hope you enjoy the video. Yeah, and probably over 3 months ago, I would have had all the faith in the world in scientists. And I still have, obviously, faith in the scientific process, as I'm sure you do, but not necessarily in the truthfulness of scientists themselves. I mean, I've made a point of getting various views on this topic. I had, before I interviewed you, I actually had, I had a guy called Mark Pellegrini come on the podcast, and he was staunchly against the lab leak hypothesis. And I'm not sure if you listened, but after our conversation, I also interviewed Professor Peter Doherty. Do you know who that is?
I'm not sure I do, but I apologize, I missed that.
He's actually a Nobel Prize winner and the patron and namesake of Australia's most highly regarded virology institute, the Doherty Institute. And even he was very defensive when I brought up your Bayesian analysis and the lab leak hypothesis. But it turns out that his institute, the Doherty Institute, received $3.2 million in funding from the Jack Ma Foundation during the pandemic, and a further $2 million from TikTok, both of which are obviously Chinese companies. I didn't bring that up with him because it seemed, I don't know, a bit indecorous or something. But I guess what I'm getting at is it's really disturbing to think how pervasive the influence of the CCP is. Just how far-reaching is their influence, do you think, in the entire scientific community?
Oh, I think they've done masterful is almost the word I would call it, but yes. So the journals, so Lancet, Nature, to the least extent Science, have largely been co-opted, is the best word to say it, by various ways via the Chinese government. At the university level, the direct grants for scientists inside American and Australian and European institutions is immense. But a second, and actually maybe more important financially level, is that, you know, the universities have this structure of tuition where for a foreign student, it's, you know, 2, 3, 4, I mean, huge multiple of the in-state or in-country tuition. And the Chinese just send as many students as can be taken. So indirectly, you're sitting there, you know, you're the CFO of Harvard or something, and you're looking at your budget and you're saying, oh God, you know, 80% of my budget is coming from out of country students from China. And so that just has an effect. And I know Australia has a bit of a problem in that. If I've seen some journalist articles around the magnitude of the foreign, specifically Chinese foreign students in Australian universities, and it just can't help but have an effect.
That's fascinating. So rather than, you'd think charging international students more to attend your university would give other countries less influence over your university, but in fact the opposite is true, because the university is then receiving 3 times as much money from those countries and from their citizens. Wow.
Yeah.
Aside from it actually gaining traction in the public dialogue, has the argument for the lab leak hypothesis gotten stronger since we last spoke?
A little bit. I mean, it was really quite strong. There've been a couple pieces of new information, including this amazing group called Drastic, of course. I'm a member of it, but most of the other people are on no. They're completely avatars, as it were. But they've come up with 3 amazing Ph.D. and master's theses done at the Wuhan Institute of Virology over the last 6 or 8 years that just walk you through all of the possible findings that you would need to have to help create SARS-CoV-2. There's not a direct tie-in, although there might be in one of them. We're still investigating. It's only been available in English 4 or 5 days. So it's just a masterful piece of work. Of course, all of the work, I think it's disingenuous when people say there's no evidence for a lab leak in the context of doing 80,000 specimens from nature and not finding a single case. That is evidence, that is data, and there's a way to interpret that data. You run it through a statistical analysis, and you say, the incidence in the world has to be less than this to get that result. And that number is so low, it's a zoonosis we've never seen before. That kind of thing. So there's that. I mean, I did a little new analysis myself that kind of, again, it's sort of a completely sort of new way of looking at it, but it points to the fact that, I mean, I can now almost time the experiment when the furin cleavage site was put in the virus to sometime around the end of August or September of last year, of 2019, excuse me.
How were you able to determine that?
So it's a bit complicated, but let me walk you through it. There's a spot on the spike protein called 614. It's nothing sexy. It's the 614th amino acid from the left-hand side of the protein. When you look at every coronavirus in nature, everyone in nature, everyone, all 58 out of 58. So that's a high probability. That's under 2%. 90% of them must have this. They all have D. They all have this same amino acid D. Okay. So all 58 have D in that position. All 58 do not have a furin cleavage site. Okay. So they don't have that. So the first patient with the coronavirus at the PLA hospital has a virus that has a furin cleavage site for the first time that's ever been seen, and a D at position 614, excuse me. So fast forward 3 generations later, that D gets changed to a G. I'm sure it was probably almost accidental. That's how evolution works. Accidents that then get selected for. So it was an accident change from D to G. It only takes one of the 3 nucleotides to do that. So it truly was a single mistake. But now the G form is 90% of all the cases in the world.
Right.
So at the same time that coronaviruses, for reasons I don't know, only God knows, for coronaviruses like a D in position 614, because they all have it, everyone has it. In the presence of a furin cleavage site, it's not compatible. So in the laboratory, what happens if you grow SARS-CoV-2 in the laboratory, it will either kick out the furin cleavage site and get rid of it, or it'll change D to G. So, okay, so once you say that, so how could a virus? So it took 3 passages in humans to go from D to G. So that was a lot of pressure on that spot. And we now know why, because it does some really crazy stuff where the spike protein stands up with the D, but it falls down with the G, and it makes it much more transmissible. So there's an explanation for why it really likes G when there's a furin cleavage site. When there isn't a furin cleavage site, it likes D for some reason. So it's literally impossible for this virus to have jumped in animals. It could only have jumped 3 times, is my analysis.
Artificially.
That's where I come to September 1st. And even in the laboratory, it gets kicked out. So I'm saying this must have been sort of introduced the end of the summer, the furin cleavage site introduced the end of the summer to a virus that had a D, because every virus in the whole world has D. There are no Gs in the world anywhere. Right. So even in a laboratory, you're always going to use a D. And then it didn't like it, and then so it very quickly it jumped to humans, and then it kicked it out. So if it had been in the lab even 3 or 4 months earlier, the first human would have gotten a G furin cleavage site, not a D furin cleavage site.
Could you just for the listeners as well remind us what a furin cleavage site is and why that's indicative of a SARS-CoV-2 coming from a laboratory rather than from a natural zoonosis?
Yep. So the spike protein has 2 main functions. The function of the first piece of it, the left-hand piece called the S1 piece, is to interact and bind with the cell to get really close, so you then can do the next step. So it's basically a bear hug with the cell, and that requires the S1 unit and the furin cleavage site. I misspoke. These two. The S2, the other part, the right-hand side of the spike protein, is responsible for coming apart, unfolding, and basically injecting like a hypodermic needle through the membrane so that the RNA can pour into the cell, take over the cell, and reproduce itself. So S1, S2. They have to be cleaved. They have to be separated in order to do that. So before the furin cleavage site was put in place, there's a site called the S2' site, which is a little bit to the right of where the furin cleavage site is, and it's identified by a couple enzymes. They're not found on human cells very often. It's a very limited repertoire of enzymes that can activate it. Because remember, the virus has to do two things: it has to bind, and then it has to be cleaved and activated to go into the cell. You can do the activation ahead of time, so it actually can come to the cell pre-activated. So the beauty from a transmission point of view, from a killing point of view of the furin cleavage site, is it opens up the sources of doing that cut that's so necessary for the infection. And so in fact, it generates a site that's recognized by furin, which is a very common enzyme in human lung tissue, very common enzyme in human nasal tissue. And so it's the perfect, as I've said in some of my Bayesian analysis, scientists in 11 different laboratories since 1992 have put a furin cleavage site in cells on purpose to see what happens. And in all cases, it's more transmissible, more infective, and more lethal. It never disappoints in that context. So, so that's the significance of having the furin cleavage site. 2,000 coronaviruses of the same class, 0 furin cleavage sites. They don't have them. And it might actually be because it is too deadly, too infective. So viruses like Ebola don't take over the world because they kill so many people that they don't get the chance to pass on.
Right. So it doesn't. So coronaviruses, when they occur naturally, don't have furin cleavage sites, generally speaking, because, well, actually 1,999 times out of 2,000, they don't, because that prevents the virus from spreading as much, because the host dies before it can infect someone else.
That's right. Yeah, it is so effective, it effectively kills the host. So there's a fine line. I don't know if you have the story of Goldilocks and the 3 bears in Australia. Yeah. So, one is
just with an Australian accent.
Yeah. Okay. Yeah. So the soup's too cold, and then the soup's too hot, and then the soup's just right. So to be a pandemic virus, you got to be right in the middle. You got to be not so weak that you don't get slammed by the immune system and wiped out, or not so deadly that you don't have time to pass it on. And the way it works in humans, of course, if you end up in the hospital, your chances of passing it on goes way down because you're in a room by yourself, you're isolated. So asymptomatic viruses are really the best. And I have another important point on that if you want me to go.
Absolutely.
There.
Yep. Absolutely. Go ahead.
Okay. So there's this protein called ORF3A in the coronavirus. ORF, open reading frame. That's science talk for this looks like a protein, but I don't have a clue what it does. So what it means is it has it has a methionine, which is the first letter of every protein. So, and then it's a bunch of amino acids after that, but nobody knows what it does. But the Wuhan Institute did a lot of work on ORF3A, and especially in one of these theses that just came translated here in the last week or so. And what they found was that ORF3A is responsible for doing something with the nuclear membrane and suppressing interferon. So it makes the cell make less interferon. Why is that important? When you get infected with a virus and you sweat and you have a fever and you're red and you're just streaming and you feel sick, like everything is coming apart at the seams, it's not the virus mostly, it's interferon. So when your body gets in a virus infection, it wants to make interferon, and because the interferon turns on your immune system and fights the virus. By suppressing interferon, you make the patients more asymptomatic.
Right.
By suppressing interferon, you reduce the immune response, so it makes it more deadly.
Right. So the furin cleavage site without the ORF, what is it? ORF8, did you say?
ORF3A.
ORF3A. Without the ORF3A alteration, it would not have spread as well as it did, because it would have killed the host before the majority of hosts before they were able to.
I think the best way I'd like to frame it for your listeners is to say, if everybody became symptomatic within 48 hours of getting a coronavirus infection, it would not have spread this way. Because when you feel as bad as you feel with a lot of interferon in your bloodstream, you don't go to work, you don't go out of your bedroom, you lay in bed, and pray that you won't die. So the fact that that was not happening allowed people to be in the community for 6, 8, 10 days, spreading it like crazy, without having symptoms. So it's more the spreading capability than the lethality.
So it's just a perfectly made biological killer.
Your words, not mine, but I don't disagree.
I keep trying to get emotional answers out of you.